Hello, everyone. My name is Sarah Jane Demy, and I am the founder and CEO of Demy-Colton. I would like to welcome you to the Demy-Colton Virtual Salon Series. Demy-Colton's mission is to convene biopharma and digital medicine industry leaders to catalyze change. We know we can help drive innovation when we bring individuals together who are united in a common cause and a shared passion. And in our industry, that cause and that passion is bringing cures and therapeutics to people in need. We were in the process of launching a new business development and investor conference, BioFuture, here in New York City when COVID-19 interfered with those plans. BioFuture is now postponed until 2021. However, we brought together such an amazing group of speakers and participants for BioFuture 2020 that we decided to continue our mission of bringing thought leaders together virtually until we can meet in person, hence the birth of the Demy-Colton Virtual Salon Series. We would like to recognize and thank all of our sponsors who have stood by us and during this challenging time as we've changed our plans and broadened the programming that we're doing. Today, we are very excited to feature the second installment of the Demy-Colton Virtual Salon Series, Drugging the Undruggable, the Power of Artificial Intelligence. We are really pleased to be joined by Robert Schneider of NYU. He will be the moderator, along with Mary Nakata of JJDC, Alice Zhang of Verge Genomics, Noam Solomon of Immuneye, and Abe Heifetz of Atomwise. And if all goes well, next year, our esteemed panelists will be joining us at our in-person BioFuture event here in New York City in April of 2021. Now, with that said, I'd love to introduce our moderator, Dr. Robert Schneider. When we first started planning BioFuture here in New York City, everybody I spoke to said that I needed to speak to Bob because he was the true mover and shaker here in the New York City community, and in fact, on a global basis for biotechnology. He is a leading biomedical investigator and has been a biotech entrepreneur for 30 years. He is a co-founding scientist of 8 biotech companies, including ImClone Therapeutics, acquired by BMS, Kanji, PTC Therapeutics, and most recently, ENB Therapeutics. Bob is the Albert Sabin Professor of Molecular Pathogenesis at NYU School of Medicine, and under his leadership, NYU has become one of the leading academic centers in the U.S. to translate biomedical discoveries into new drugs, medical devices, and other products. And even more importantly, Bob has been a mentor and an advisor to literally hundreds of companies that have been launched here in this local ecosystem, and it's his passion. Bob is also the co-author of more than 180 peer-reviewed publications in leading journals and has received multiple honors and recognitions for his work, including most recently the 2019 American Cancer Society Cancer Researcher Award. I'm really pleased to turn the mic, so to say, over to Bob and his esteemed panelists. Bob?
Thank you. It's a pleasure to be able to moderate and host this today, and I welcome all of you that are joining us. I understand we have well over 400 people that are now attending this webinar. That really speaks to the importance of artificial intelligence and the ability to utilize that in multiple platforms to build life science companies. So good morning, good afternoon, good evening, and good middle of the night. I know that there are people attending from all over the world, and so it's a pleasure to be able to do this. What I'd like to do is, with our panelists, develop the entire range of the uses of artificial intelligence. We'll start by discussing exactly what is artificial intelligence. We'll discuss its multiple indications. It's actually very broadly applicable, but a bit of a mystery to many people. It's actually not a black box, and we will actually take it out of the box this morning and talk a bit about how AI can actually be utilized in so many different aspects of development from drugs to identification of patients. There's so much that it can be used for. And so we're really representing that in our panelists this morning. Now, we can't develop drugs using AI if we don't actually know how to bring in the investments to do so. And for that reason, we also have Marianne here and field quite a bit of questions and discussion about how one actually creates the financial structure to develop an AI company and develop drugs in this fashion. Quite a bit different. And so we'll try to cover all of that. And so with that, it's time to begin. So let's start by having each member of the panel introduce themselves. I'd like you to talk a bit about your companies, an overview about how AI is used in drug discovery or drug development or patient identification and what you do. And then we're really going to dig in. So if we could start, Alice, thank you.
Hi, thank you. I'm Alice. I'm the CEO and co-founder of Verge Genomics. And at Verge, we're using machine learning on human genomic data to develop new drugs for complex diseases like ALS, Parkinson's disease, and Alzheimer's disease. And I started Verge really a few years ago out of a frustration with a lack of human data that's being used in drug discovery. And I saw that drugs are being developed from the wrong targets that were tested in animal models that ultimately weren't translating to humans. And I also saw that we're in this incredibly unique moment in time right now where advances in human genomics and machine learning have given us the tools to generate human data at a scale that wasn't possible before. And so I started Verge with the vision of developing a next generation therapeutics company that could take all of this human data to unlock a spectrum of these diseases that have been considered traditionally intractable, such as ALS, Parkinson's disease, and Alzheimer's disease. And so using this approach, we've been able to go from concept to our first development candidate in the last 2 years. And our first program is in ALS. And that's a small molecule, which will be entering clinic next year. And that's always been a big dream of mine, is to see some of these methods actually bear fruit in terms of real products for patients. And we've also established a pipeline of over 4 different programs that will be entering clinic in the next 2 years. And lastly, we've also partnered with over 21 global academic centers around the world to create one of the field's largest proprietary databases of human brain tissue. And we've RNA sequenced over 6,184 patient brains that have been transcriptomically profiled. And so it's one of the most ambitious and fulfilling missions I've ever worked on, and I'm excited to talk more about it.
Great. Thank you, Alice. We're going to dig into that quite a bit and really try to understand how the power of AI has accelerated your R&D efforts and the ability to get into the clinic actually so quickly. And it's really quite remarkable. Noam, can you tell us about yourself, your company, which is actually quite a different application of the use of these AI tools?
Yeah, definitely. So I'm Noam. I'm the co-founder and CEO of Immunite. We started just a year and a half ago, and we just broke out of stealth with the fundraising round that we announced recently. Our company is concentrated around discovering and better understanding the immune system by using single-cell sequencing and methods with AI. I'll be happy to explain more about the AI part, but we are building the largest proprietary database for single-cell sequencing data around the immune system, especially around the peripheral immune system. And we're working with more than 12 academic institutions and hospitals, and we started a few industry collaborations with biopharma and biotech companies.
Thank you. That's great. And we're going to dig into that as well, because you're using AI in the course of single-cell genetics, especially for mapping the immune response as well. It's a very different type of application. Abe, tell us about yourself and your company.
Thanks. My name is Abraham Heifetz. I'm co-founder and CEO of Atomwise. We invented the use of modern machine learning for drug discovery. So we're a structure-based small molecule drug design shop. And we were the first ones to use modern machine learning techniques for that problem, with the goal of giving medicinal chemists superpowers. So our species' best technology for problems in image recognition and speech recognition is something called deep learning, and specifically convolutional neural networks. We were the first ones to realize that you could apply that to molecular recognition. And these are techniques which probably most people in the audience actually have used quite a bit. If you've ever spoken to Siri or Alexa, if you've ever uploaded a photo to Facebook and it said, "Did you mean to tag your friend?" These are, you've used a convolutional neural network. And so we applied that technology to drug design for the first time. And then we've actually gone and been running the biggest application of machine learning to drug discovery in history, with over 750 projects that we're working on across 600 unique targets, working in 40 countries. And we've been able to deliver a success rate of finding compounds that are interesting to move forward with over 75% success rate.
Great, thank you. We're also going to spend quite a bit of time discussing how we actually do that. It's a bit of a mystery to many people who don't work in this area, how one actually develops and applies artificial intelligence in all these different formats. And so we're going to get back to you on that as well. And last, Marianne, none of this is possible without people like you that actually develop the financials and fund these companies. Tell us a bit about yourself and what you do and who you work with.
Sure. I'm Mary Nakata with JJDC, Johnson & Johnson's corporate venture group. We invest in all three sectors that Johnson & Johnson has, which is consumer, pharma, and device. We are strategic investors, so we require co-championship of the folks in the R&D business for us to make investments. We invest at all stages, from seed to Series A, B, and beyond, and we'll actually do investments into publicly traded companies called PIPEs to support business development deals. We lead investments, I'd say almost a third to half of the time. And but we don't have to lead the investments, and we write any check size, and we feel that this is an important part of a deal structure that could also involve, let's say, a license, a collaboration, or just a pure investment. And we invest globally and have 12 investors deployed in San Francisco, Boston, London, Shanghai, and I'm based in Boston.
Great, thank you. We're also going to spend some time talking about how one actually begins to raise funds, whether it's seed or Series A or B, when one utilizes AI technologies, how one does a pitch in that area. It's quite a bit different than a lot of the conventional approaches that companies use actually for development of small molecules. Great. So let me ask a question for all of you, and we can start in any order. But what is AI? That's really a black box to many people. And how is that different than traditional statistical analyses? And what is artificial intelligence? Is it really different than things like causal machine learning? Can we define that for people? Anybody? Abe?
Sure. I think these techniques are closely linked. One could take a cynical view, and there's a quip that I've heard, which is, when you're fundraising, it's artificial intelligence; when you're hiring, it's machine learning; and when you're implementing, it's logistic regression. And so even the framing of that joke, shows that these are closely related techniques. I think most of the progress in modern AI is in statistically based machine learning. And so I think it's reasonable to think about these things as highly related. Mm-hmm. I think the important difference to highlight comes really from Hegel, which is changes in quantity lead to changes in quality. And modern AI approaches, I think the interesting thing about them is that they have yielded substantively different levels of performance. What we've seen is transhuman performance, beyond human performance in problems people worked on for decades. Obviously things like chess, obviously things like Go, but also things closer to this discussion, diagnosing diabetic retinopathies in patients and so forth. Yes. And so I think it's what the AI has been able to deliver. At Atomwise, we've been running the biggest screens in human history, like in terms of diversity and size of small molecules. These days, you can have 16 billion molecules that we can do in about 2 days, and that hasn't been possible before.
That's great. Thank you. No, you use AI and develop AI in a very different way. Can you describe that a little bit to us as well, and how that's different than standard statistical tools?
Yeah, sure. Maybe I'll say one thing that when people say AI, they usually mean different things. I think maybe the broader definition of, or the broadest definition of AI is making machines behave intelligently. And the definition of intelligence is something that you can really bring different types of substances. But I think today, 2020, we are kind of in the middle of a perfect storm where you have machines that are incredibly strong. So the compute power is very, very strong. You have very interesting framework, different machine learning models, deep neural networks, the classical statistical machine learning models. And then there is the data that becomes available. In our area, we're talking about biological data, or in my area, it's sequencing data. When you combine all three, you can talk about how you can leverage AI to bring some intelligence. Mm-hmm. And so I don't view AI as a separate thing. I think that AI can empower us to make better informed decisions. And at Immunai, what we are doing is, as I mentioned earlier, we have a lab in New York City. Actually, you know where it is, Bob. And we are sequencing thousands of samples with techniques of single-cell RNA-seq and other types of sequencing that we sequence samples on the cellular level. And for every cell, we can see tens of thousands of different genes, hundreds of different surface proteins, clonality data, and more. And then the question is, what do you do with the data? Now, RNA data, gene expression data, is extremely noisy. So what you're trying to build a database, because in big data, you can apply complicated machine learning algorithms, but then the data is noisy. Yeah. So first application of AI in our case is to clean the data, to normalize the data, to deal with maybe the most difficult problem in computational biology, which is the prohibitive batch effect problem. And that's what we're doing as a first step in applying AI.
Mm-hmm. Great, thank you. So let's move on a little bit, and let's talk about some of the applications of AI. And Alice, perhaps starting with you, because you started with as a company where you sort of knew your targets, but you knew that they were not druggable. And one of the reasons so many people have tuned in this morning is because of the power of AI to be able to tell us how to drug compounds, how to drug proteins that are formerly undruggable. So if you could tell us a little bit about how you've used the applications of AI in that regard, and then also how your large databases are helping inform your development.
That was addressed to me, or
yes.
Abe?
Yeah, to Alice. Yeah.
So one of our big realizations is that many of these diseases, like especially in neurodegeneration, many of the failures were resulting from selection of the wrong targets because these are incredibly complex diseases. And so what's really changed in the last 5 years, in our view, is our ability to generate the right data at sufficient scale. And so what we do is our belief is really that in order to succeed in humans, you really need to start in humans. And what we do is we start from day one with a large proprietary database of human brain tissue. So these are over 6,000 human brains we've sourced from around the world from over 21 different hospitals or brain banks that we then internally RNA sequence and combine with large-scale human genetics to identify novel targets. And what's important is that we also have the back end of our own in-house drug discovery labs where we have human-derived neurons that we can then continue to de-risk and validate these human-derived targets from, to the goal of creating first-in-class molecules that have a greater probability of translational success because we're going from human to human rather than from animal to human. Mm-hmm. And so a lot of our focus has been mostly on the generation of the right data and sufficient data from the tissue samples from patients themselves to identify targets that have really strong linkages to the human genetics, which has been shown to more than double your chances of clinical success.
Mm-hmm. So perhaps in a little more detail, how do you actually do it? How do you actually develop AI in a way that leads you to a target, and how do you verify that?
So those are both good questions. And what we use AI in a whole number of different ways. And when you ask what is AI, that's a great demonstration of even within the same company, we use a ton of different methods. And so one of the first methods we do is we have this large database of human tissue, and we use this tissue to identify gene signatures or groups of gene networks that are consistently dysregulated in patient tissues, but not in age-matched controls. The next challenge is how do you actually figure out which one of these pathways are causal versus just the body's downstream reaction to the disease. And this is when we use a second different method to integrate the genetic data with the RNA data. And that's been a key discovery for us, which is that looking at any one type of data has not been particularly helpful, but it's really through integration of different data types that we start to see convergent signal that really is the smoking gun for biological mechanisms. And what we do is we then look upstream to identify causal intervention points that we can drug using small molecules or gene therapies with the goal of reversing or normalizing that patient signature.
How do you get these different areas of statistical analysis to talk to each other? Right? So you're moving through biology, through systems, networks, all the way to protein topography, protein structures to determine what's druggable. How do you integrate all of that? And that's really the power here.
Yeah, that's part of the secret sauce, I'd say, of what is proprietary at Verge. I think a good metaphor is it's similar to baking a cake. All cakes have the same basic ingredients: flour, egg, water. It's what ratios you use them at that determine whether it's a good or bad cake. And similarly, it's what are the right parameters to use? How do you actually truly integrate these different algorithms for the different data types in a way that yields biological signal? And then importantly, how do you actually verify it? So for every single prediction that we make, we generate a huge amount of validation data in our labs that actually feeds back into the platform itself and continues to retrain the algorithms to improve over time.
Yeah. And really applying the same issues and questions, Noam, to your company as well. You're doing the same thing in a very different way with respect to training our understanding of the immune response. Could you describe a bit about how you've been able to employ these types of tools?
Yeah, sure. So as I mentioned earlier, so there is this database that keeps growing and you need to normalize it. And we use to the normalization of this database different types of machine learning algorithms. One of them is encoders. Once this data is what do you feed the machine? And in our case, we are disease agnostic. So we take sample from variety of indications, many cancer indications, autoimmune indications, Alzheimer's and others, because our algorithms, or at least some of the framework that we use is called transfer multitask learning. So we are trying to identify and isolate the immune component in a disease and transfer insights across indications and therapies. And so I'll give an example to be a bit more concrete. There are algorithms that are in the area of computer vision that you can train neural networks to detect a dog in a picture. So you feed it many different tens of millions of pictures, and then the machine can say, it's a dog, it's not a dog. And then you take the same neural network and you want to train it to identify a cat. So a cat is not a dog, but there are many similarities. So you can transfer this model and train it on, let's say, 1,000 different pictures, not 10 million pictures. So similar and non-small cell lung cancer. Maybe you can transfer insights from melanoma to non-small cell lung cancer, to breast cancer. And that was the starting point for us. How do we build the right database? And from very early days of the company, we were able to see that we can transfer insights from autoimmune disorders to cancer and across different indications. And yeah, and that's a lot of the AI that we are using internally.
How much of the AI that you use, and this is really a question for all of the panelists, how much of the AI that you use is actually public domain? How much of it do you have to develop in a proprietary way? which really gets to some of the questions that people have about the utility of AI. Do you need to have that kind of understanding and expertise in-house entirely?
Everything that we...
Oh, sorry.
Go ahead.
Go ahead, Dave.
Go ahead, Dave.
Everything that we use, we built internally.
Yeah.
And in some sense, I think it's like, if you have an assay, how important is it that you know how the assay works, right? Like, how important is it to know the details if you're going to build it? There are some kits, but even with kits, it's very useful to really understand if you're going to get clean signal out, what went into it and what worked. When we started, you asked how different people's AI worked, and let me say, I think the question about how AI can drug the undruggable is exactly the right question. If the only thing that AI would let you do is come up with a drug for a kinase that people have 1,000 drugs for, right? That's not going to be very interesting. If it's you have to have a set of known drugs and it gives you the methylated form of those drugs, probably you don't need AI. Probably any medicinal chemist could do that for you, right? And so I think the whole question is how AI can push into previously undrugged biology. And in some sense, that's what gets us excited about any new modality, right? Like the reason why we're interested in CRISPR or in protein degradation or in gene therapy, like is because it gives us a handle on these new previously unexplored dark areas of biology.
So one of the common areas then that comes up then among all of you is that the real power of AI in drugging the undruggable is to be able to integrate multiple different types of big data and networks to actually arrive at appropriate targets, more so even than being able to design molecules, which it can be used for as well.
My perspective, and the one that we take at Atomwise, is if you identify a target, and I absolutely agree with Alice that if you identify the wrong target, it doesn't matter what you do down, further in the pipeline, you're not going to get a drug out, right? And so target identification is of course critical, but it's also an incredibly challenging task. And we have many targets which have been implicated in human disease. If you go on the Human Protein Atlas, actually 4 times as many targets for which we have an FDA-approved drug have been implicated in disease and have been undrugged. We have good evidence that we would like to be able to do these. And these are the ones that people just know the names of, KRAS and c-Myc, and I mean other broadly known ones. And so there's gold in them dar hills if we were only able to figure out a way of interceding in the biology. But the other piece is, if the cost of evaluating one of these targets, the cost of developing a drug and proving that it works, if the cost was $1, you would try all the targets, right? Your ability to choose the target correctly is intimately linked with the effort it takes to interrogate the biology. So if we can drop the cost of developing safe, effective treatments against a target by a factor of 10, then we could be 10 times worse in identifying targets and still have the same kind of output.
So then, speaking to that point, how are the different ways that AI is actually used to accelerate drug development, drug discovery? We've touched on some of that, but what's the evidence that AI is actually very effective in this regard? And how do we validate that?
Yeah, at the risk of speaking for my panelists, but I'll give you a quick example, which is it's a nice head-to-head. And so one of our commercial collaborations, so I won't name the protein, but we were going after a kinase. But it was an interesting and challenging kinase where there were two very, very closely related kinases, the 2 amino acid difference in the active site. And if you hit one of the kinases, you would cure people's cancer, but if you hit the other one, you would kill those people by giving them heart attacks, right? And so the entire challenge, for this particular protein target, the thing that made it undruggable was selectivity rather than finding a molecule. And so this was a protein target which lots of big pharma companies, small pharma companies, AstraZeneca, Roche, Janssen, others had gone after, had failed to deliver selectivity. And so although some of those compounds had gone into the clinic, they were struggling. They were failing because of unacceptable cardiotox. And so our collaborator, our biologist, said, look, we know that this biology is interesting. We know what the problem is. And it's a question of developing the small molecule which would unlock treatment for patients. And so they said, if your AI hocus pocus could deliver that, that would clearly be valuable. So we ran a massive screen. One of the incredible things about medicinal chemistry today is actually that 99.99 something molecules which are available to the chemists today don't exist and have never existed on this planet. Like the chemical manufacturers have embraced synthesis on demand. And so that means you can get any of 16 billion molecules delivered to you in 3 weeks, ready to go into your assay in 3 weeks, 6 weeks. But most of those, no one's ever built before. They just think they can build them. So we did a large screen and against this kinase pair. And in less than 6 months, we had the first selective inhibitors. So that's an example where head to head, right? The big pharma had every tool, computational, physical screening, high throughput screening, human intuition. They'd all been brought against this challenge and failed to deliver, but AI succeeded. And
AI succeeds in a way which is proprietary for each of your companies as well. And I think that's one of the key points. So how do you find the talent? How do you find the computational people? How do you integrate those people throughout the rest of the teams in your company? Everybody speaks a very different language when they're doing computational analysis and AI than do the traditional biologists and chemists. And that's a real challenge. So how do you build a company where AI is really the foundation of your company and integrate that? And maybe Alice, starting with you, because you've done this.
Yeah. I've had this has been one of my biggest focuses in the last few years I've been running the company, which is that what I learned pretty quickly was that one of the greatest silos, or one of the greatest barriers to fully realizing the power of AI in drug discovery, were these silos that I saw existing between computational teams, biologists, chemists, drug developers. And in the first year, I had to interview probably now over 1,200 people, actually just to build the core team that we ended up getting. And what I learned from interviewing so many people was that what was interesting was I saw a lot of the best AI and computational talent was still within academia. And that we had to put them side by side with drug development veterans and neurobiologists and chemists to really get them to work together. And the second thing I discovered was that a lot of what was attracting people to our company and also retaining them was the unique opportunity to really be in the driver's seat of drug development as a computational biologist. I think it's very rare that you have a company where the computational scientists are driving which targets we pursue, how we invest our resources, what are the drug development programs we initiate, and at the same time also investing in generation of cutting edge data and new methods. Because I think what you see frequently in a lot of companies is that they're often in a support role for other therapeutic area heads. And that's combined with, I think, the focus on disease area, neurodegeneration. So about half our team has a personal connection or a family member that's been affected by neurodegeneration, have kind of been the three cornerstones that have helped us attract and retain a lot of computational talent.
So, Noam, you are in the process of building a company along this way. Can you tell us also, how do you find the talent? How do you integrate them? Your company at this point is much more AI-based and less drug discovery-based, although it's moving in that direction.
Yeah, it's a great question. First of all, I think I was very fortunate to found the company with my CTO and co-founder, Louis Volok, who won the best thesis award in machine learning at MIT. And he also worked in Palantir later. And so from the beginning, we knew how to build and how to interview, how to screen for the best machine learning talent in the industry. And like Alice said, we were fortunate. First of all, I think that I don't define us as an AI company. We are a biotech company. The bio side is as important as the tech. The AI in our company is kind of split into the machine learning heavy mathematicians coming from the top research institutions and the top tech companies like Facebook and Google and others. But also we have an amazing team of computational biologists and wet biologists and immunologists that help build this thing together. We think that in order to implement a super strong AI team, you need to have a very strong engineering pipeline in the company. So it's not, and that's maybe the difference. It's not like we're not building MIT or Stanford in the company. We are bringing people from MIT and Stanford and Berkeley and others, but we are also putting them side by side with the experienced engineers. And it's not just about inventing or coming up with the algorithms, it's implementing the algorithms in the most efficient and targeted way. And I think that the beginning was very challenging, and it was every top talent we really had to fight and travel all around the world to bring. But over time, we have an amazing team, and people are heard about us. They want to join, and yeah, I think now I'm fairly optimistic that this blend of computational people that are both trained in industry and academia with the wet biologists and immunologists are really working together side by side in the right way.
So Alice touched on this also, and I'd also like to ask you, how do you find talent? There's a lot of talent in academia, but they're not prepared yet for a more team-oriented corporate setting. How do you go about that? And then after you address that, I do want to move on to talk about how you get all this funded.
Money.
So your company is much more AI-based as well.
Absolutely, but I think I couldn't agree with Alice more about the need. One of the advantages, the absolute need to succeed in this, is to be cross-disciplinary and non-siloed. So certainly we employ people who have PhDs in computer science and in machine learning specifically. The person who runs my machine learning team actually was running the machine learning team at Amazon A9, ranking and rating molecules and ranking and rating books. What are the best 10 books to recommend you, and what are the best out of 100 million? What are the 10 best molecules to test out of 100 million? There are certain algorithmic similarities between those, and so it's important to have top talent in AI and machine learning. That said, we also have PhDs, we have medicinal chemists, we have structural biologists, we have physicists, we have pharmacologists, just across the entire board. And one of the ways, like one of the absolute critical things you need to succeed is bringing them together and having them fruitfully talk to each other, because these are groups with different languages, different terminology, different assumptions, and so forth. And so I think it's not just world-class expertise in one discipline, but actually cross-discipline. Yeah. And I think hiring is always very, very tricky. And so part of it is part of our good fortune was to be early in the space, to publish a lot of. There's this difference in culture, which is a lot of machine learning people expect to publish and open source all their algorithms. And that's been very strong. And actually, even in industry, you see Google and Facebook and others opening it. Whereas early publication open sourcing is very unusual within pharma. And so actually on the AI side, recruiting is helped by going to conferences and presenting our work. With pharma, it's trickier.
Mm-hmm. Thank you. I'd like to move the discussion a bit at this point to how do we actually fund AI-based companies? And so, Marianne, if we could begin that discussion. Quite a few people who've tuned in this morning are either investors or people that are developing their own new startups in this space. So let's discuss a bit about the investment strategies, and then I'd like to go back to Alice, Noam, and Abram as well to talk a bit about how do you actually raise money for an AI-based company. How do you value that, and what are the opportunities? What are the red flags to you as an investor?
Yeah, so maybe I can start. Similar to the differences with, let's say, pharma investing versus device investing, it's healthcare, but it's a different flavor. And so some of the differences are, what is the value proposition? Because when you invest in a more standard biotech that has a platform that pops out drugs, those drugs have a value that is easy for potential licensors or acquirers to define, because they can have an EMPV. This is the target product profile, this is the peak year sales of this drug, et cetera. This is the cost it'll take to develop, and these are the revenues that we'll be getting from that drug. So the analytics are much more straightforward. The value proposition for AI companies, it depends on what that company's business model is. And I think that's and so being able to evaluate the value of that business model, if you are purely focusing on R&D efficiencies, if you are defining biomarkers that will help for defining patient inclusion or the biomarkers of efficacy, how do you monetize that, and or how do you value that from an analytics perspective? And what is the value proposition? Is it a service? Is it a partnerships that will drive the revenue? What will drive the revenue of that company? And then also, I guess feeding into that is, from an investor perspective, what is the exit? Where's the liquidity? Some of these companies go IPO, and they've done that successfully. Others have the potential, I guess, to go to M&A, and that could either be to a pharma. I mean, one of the classic examples people point to is Flatiron being bought by Roche, or it could also be combined with other complementary companies. Like one might have the analytics, another might have data, so you could do a roll-up and be able to form, a company that has multiple AI value propositions. So it is definitely different, and the investors are therefore different too. There was a time, and there still are certain firms that actually have both, that do healthcare standard pharma and biotech investing, as well as AI and data science investing. But some of those firms have split because the nature of those investments and the value proposition and the return on that capital is very different. Some years ago, for example, Atlas Ventures and Cambridge split into two separate groups. So, and then I would argue, even within firms, there are investors who focus more on the AI digital side, and other investors that focus more on the asset platform-based, more standard biotech.
So if you're a new startup company, how do you know who to pitch to? How do you make those determinations, and what should your pitch look like?
Yeah, no, great question. I mean, obviously, on the websites, oftentimes the investors will show what their portfolios are. Another way of doing it is to find companies that are similar to yours, and look at the investors and the board members for those companies. And that's a great way of defining what might be a good fit for your company. In terms of the pitch, one of the most important things is to really be very clear about the business model. So we see a lot of pitches that will say, this is what the offering is, but it's not clear where they are in terms of looking for revenue, partnerships, and what their ultimate goal when they grow up is going to be in terms of, for example, M&A, IPO, that liquidity piece. Another important thing is the use of proceeds, and making it really clear, if we're asking for 20 million, what value inflection will that get you to? And so those are some important aspects, I think, of the pitch.
This is really a question for both you and then Abe has many, many different types of partnerships. What type of partnerships do you develop? Are you a service company? Are you a drug discovery company? How do you value those partnerships? If you could fill in a few of those details. In many cases, your partnerships, and this is true also for Alice and Noam, your partnerships are based on a value prop that you have an extraordinary database that's going to continue to yield new targets over time. So how do you assess that? How do you build those types of partnerships?
So for us,
Alice,
I mean, I think what's interesting is that all of our companies are kind of at this intersection of tech and life sciences. And so in that way, looks quite a bit different from other traditional biotech companies. And I think this is an increasing pattern that you have companies that are kind of fluid between these fields. For example, there's been an acceleration of new technologies, such as new modalities, ASOs gene therapy, new discovery tools, iPSCs, and new technologies, including machine learning and genomics. And so how we think about it is that our platform itself, it's disease agnostic and it's modality agnostic. However, in terms of what our internal resources towards, that's more of a strategic decision, and we have to be focused there. And for us, we've determined relatively early on that all of the value for us is in developing the right drug and developing a portfolio of drugs that we can move into the clinic. And so in that way, we look a lot more like a therapeutics company. But we have a whole host of opportunity. For example, we have the surplus bullpen of these targets we've genetically validated for which there isn't known molecules or a clear chemical path forward that have garnered a lot of interest from gene therapies or other modalities that we can then partner. So we typically partner along either modalities for undruggable targets, or we partner around entire disease areas for which the clinical development path is relatively complex. So for example, Parkinson's disease is an area where we would partner to identify targets and to continue developing them preclinically, and we would partner with a larger company that has the clinical infrastructure to rapidly move multiple programs forward.
Mm-hmm. And Abe, you also have done many, many different types of partnerships. You work with a great number of different companies. Tell us a little bit about how you structure those and value those as well. And especially if you're a new startup, which you're not, but if you were a new startup, how do you actually develop a value prop for some of this?
Yeah, absolutely. And that's a critical question because without a sustainable model, it doesn't matter. You could have a scientific breakthrough, but if you can't get that into people's hands, the world won't know and the world won't benefit. And in fact, I think this discussion is really good. I had a follow-up question for Marion in that, we were talking about AI and partnerships and that there are companies that could use it for their own pipeline. There are companies that could use it for R&D efficiency and how one articulates value prop. And I'm kind of curious from that perspective about whether it's different from any other platform, any other assay technology. If a new DNA encoded library company comes in with a breakthrough, it seems to me that there's analogous questions. And so I think for us as well. We've done big pharma partnerships and small pharma partnerships and academic partnerships. With big pharma, I think they look pretty typical co-development where there's always a handful of exciting targets that the world is interested in making huge progress on in a short amount of time. These are the kinds that we've all heard the names for, right? Like IDO and Sting and KRAS, right? I mean, like just to pick a couple. And everybody's interested in those, and so big pharma wants to make progress, especially if they've been historically intractable. The way we look at small pharma partnerships actually is we have a platform to do medicinal chemistry faster and successfully. But I think it's the case that small molecule medicinal chemistry can be brought to bear on a huge range of diseases and a huge range of protein biology. And nobody in the world, no organization in the world can be the world's experts in neurodegeneration and immuno-oncology and infectious disease and metabolic disease, right? Like, that's too bold a claim. And so by partnering with small pharma companies, we can collaborate together and set up joint ventures where we do partner with the world's experts in biology. And so they can be the experts in biology, and we can provide that superpowered medicinal chemistry, and then together succeed at this. That's the way we look at it.
Thank you. And Noam, you're the newest kid on this block. You've just gone through this with a very successful Series A round. Can you tell us how you actually develop your value, what you actually pitch, and what type of people you spoke to as well? There's a lot of interest out there, especially in people tuning in right now that are developing their own companies. How do you actually balance all of it?
Right. Yeah. So I think that our vision is a platform company. We are trying to really better understand the immune system, but there should be something concrete that we offer, and there is. So in the partnership that we seek with the large biopharmaceuticals and the smaller biotech, we actually look for partnerships. Of course, there are different types of partnerships, but this platform that we are developing, we say that we can scan the immune system and look for immune response for different therapeutics. And that's a very bold statement. What does it mean to scan the immune system? So what we say is, if we can explain to you the pharmacodynamics of your drug, what does it mean for you? What are the biggest questions? What are the biggest roadblocks that are between you and getting it to FDA approval? And there are different challenges. One of them can be repurposing of an existing molecule. Another can be, how can you slightly change it to get a better molecule? And we have different types of partnerships with different types of partners. And we also have many academic collaborations where we are trying to figure out new pathways and new immunological insights that can benefit different indications and different diseases. But ultimately, our goal will be to do drug discovery and early drug discovery, so pathways and for new targets.
Okay. So we have to start wrapping it up. I want to thank everybody. As we're wrapping up, just very quickly, how do you deal with the cynicism that AI is just another means to raise money? And just short answers, because we do need to wrap up. Yeah. Has it proven that?
More criticism.
Okay.
I think the cynicism is hard won, and I think the burden of proof is always on us. The last paper that my co-founder and I wrote together actually is about errors in all the existing benchmarks that we looked at for assessing computational chemistry algorithms, and it turns out that these benchmarks, in our opinion, are wildly optimistic. And so our sense is you have to go and demonstrate that these things work over and over and over again. And the nice thing about this industry is that we're scientists, and scientists are willing to be persuaded by data. And so if you can show, as Atomwise has, the ability to go after and succeed at targets that other people have repeatedly failed at, then people get persuaded.
Thank you all. Thank you for participating. Thank you all those for tuning in. I don't know if Sarah has some additional comments.
Yeah. I do. And I think the very first thing I want to ask Bob is, when you had to get up and leave your seat, were you looking for a bear?
No.
For everybody who's listening, Bob is up in the country, and he reported that there was a baby bear outside of his windows last night when he came home. So I thought there was a bear sighting. That may have happened. The
company showed up just in time to start cutting down a tree right outside. I had to run out and tell them.
Oh, okay. A little bit more mundane. And I really want to thank everybody for participating. I think this was a really kind of eye-opening discussion for everybody, and the different flavors of AI and how one approaches drugging undruggable targets. So, Bob, thank you as always, Mr. New York City Biotech, if you will, for leading this discussion. Abe, thank you very much for joining us. Alice, I look forward to meeting you in person. I know everybody else, but not you yet. Noam, the same. Marion, thank you very much for participating. And I know you're not really cynical, are you, about AI? No. So Marion is really great to talk to. She's a a wonderful resource for people that are looking to expand their horizons, if you will, in biotech. I'd also like to thank all of our sponsors. I know I noted that before, but they've been troopers sticking with us, through this challenging time. And I also think because they really believe in the power of the people that will bring together for our meeting called BioFuture, which will take place next year. As you're signing off, please do participate in the sign-off survey and do check the Demy-Colton website for our next virtual salon, which I believe is scheduled for July 1. And then we have another one scheduled closer to the end of July. We're planning to do them about every 2 weeks. With that said, thank you, everybody. Thank you very much.
Thank you all. Take care.
Thank you both, everyone.
Be well.
Bye now.